Hi guys, I just wanted to give you some paper 2 IGCSE Edexcel biology advice. So these papers begin with a kind of comprehension exercise. Now my advice would be to bizarrely do that at the end. So go from question 2, answer the paper, and then go back to question 1, because then you'll know how much time you've got left and you can actually spend time reading it properly, answering the questions properly. Whereas if you kind of dive in, you're more likely to skim read it and rush the first question. So personally, I always advise my tutees to start question 2, work through the paper systematically, and then go through question 1 properly, and you'll tend to find that you have plenty of time to do that. I will go through this particular paper, but I'll do it at the end. Right now I'm going to focus on some paper 2 content, so I'm going to be giving you some of my perfect answers for the paper 2 content, so that's the bold part of the specification. So I'm not going to go through every single point, I'm just going to go through the points which I think are most important. So we'll start by looking at protein synthesis. Now remember that a gene is a section of DNA which codes for a particular protein. And so DNA is made up of 4 bases, A, T, G, C. So they stand for adenine, thymine, guanine, and cytosine. And remember that your base pairing rules mean that A always binds to T and C always binds to G. So straight letters together and curly letters together. As well as the bases in DNA, you also have a sugar phosphate backbone, and that a sugar followed by the phosphate followed by the base, this is known as a nucleotide. So remember, the first step is transcription, and this, remember, occurs in the nucleus. So what you find is that the DNA unwinds, the bases are exposed, unpaired, and notice that it's mRNA this time. Bases pair up with exposed DNA bases, forming a single-stranded mRNA molecule. And notice that RNA has no thymine, so uracil is used instead. So if we try and look at this in a bit more detail, if I draw the nucleus here, so here's the rest of the cell, say we unwound some of the DNA strand to get a single strand which looks like this. Now the mRNA bases floating around inside the nucleus, they come and pair up, and so what the base pairing rules would consist of is C binding to G, G binding to C, and instead of A binding to T, as would be the case in DNA, instead it's replaced with U. T binds to A as usual, A binds to U again, C binds to G, and G binds to C. So this here is your single-stranded mRNA. And to be honest, once that mRNA has been formed, it needs to leave the nucleus. So we're ready to move on to step 2, or stage 2 of protein synthesis, which is known as translation. And this occurs on a ribosome, which is why we say the function of a ribosome is protein synthesis. So I'm going to draw the ribosome here, and I'm going to attach an mRNA strand, and I'm just making this up out of my head, but it doesn't really matter what it is. So here's our mRNA. Now notice that when you have 3 bases together, such as this one, this is known as a codon. Now floating about inside the cytoplasm are tRNA molecules, and crucially they have amino acids attached at one end, and at the other end they have an anticodon which is made up of 3 bases, so we call this the anticodon. Now that anticodon has to be complementary to the codon. So for this tRNA molecule to be brought along to this particular part of the mRNA, then the anticodon would need to be A, U, C, because these would be the complementary bases. So at that point, this tRNA molecule would be brought here and it would bind, bringing with it this amino acid. Let's look now at the next codon, CCA. So this time we need a different tRNA molecule. And this would have a different amino acid attached. And so the anticodon this time would need to read G, G, U. So in this case, the second amino acid would be different to the first. And as you can see, we'd be building up a chain of amino acids which would join together with peptide bonds to become the protein. So I'm now going to summarize that down here. So translation, as I've already said, occurs on the ribosome. mRNA attaches. tRNA, so that's transfer RNA, brings amino acids to the ribosome in the order dictated by the codons on the mRNA molecule. Amino acids are joined by peptide bonds to form proteins. The next crucial part of the spec I want to talk about is the kidney, because that's a real paper 2 topic. So it's so important that you know the structure of the nephron. So I'm just going to draw a very rough diagram here. Be prepared to label this. Now remember that the blood vessel entering the nephron is known as the glomerulus. And the vessel entering is wider than the one leaving, which means that there's a buildup of pressure here. So let's get labeling. So this is the glomerulus. Here we have the Bowman's capsule. The first coiled tubule is here, which you can call the first coiled tubule or the proximal convoluted tubule. Either is fine. They both mean the first coiled thing. Because this is also coiled, it's known as the second coiled tubule or the distal convoluted tubule. And then here we have the loop of Henle or Henle. And then we have the collecting duct here. Now some very important processes take place in the kidney nephron. Number 1 is ultrafiltration. And I've already said that up here, this vessel is wider than this one, which creates a pressure, and that pressure forces small molecules out of the blood into the Bowman's capsule. So that's why ultrafiltration takes place here, and it's always good to label where these processes take place. So ultrafiltration, small molecules, and let's name them, e.g. water, urea, which is the excretion product of the kidney, ions, and glucose, are forced out of the blood into the Bowman's capsule from the glomerulus. So that's a nice full answer there. And then number 2, selective reabsorption, actually takes place here, and that's where some of these small molecules are returned to the blood, so they leave the nephron. So number 2, selective reabsorption. You've got to be clear as to which molecules are moving. All of the glucose and some ions are returned to the blood from the proximal convoluted tubule. Notice that this occurs against the concentration gradient, so it's done by active transport. And let's state what that is for a very full answer. So they move from an area of low concentration to an area of high concentration, and therefore it requires energy or ATP. One thing I forgot to point out up here at ultrafiltration, you must be careful. Notice that proteins do not enter Bowman's capsule, and that's because they're too big. So we've discussed a large part of the nephron in great depth, but now we need to look at the collecting duct, and really we're looking at controlling water content of the body. So this is called osmoregulation, so control of water content. And we need to understand all the steps in two scenarios: when we've had little to drink and when we've had lots to drink. And we can build up our answer nice and easily. So first of all, osmoreceptors. And where are they? In the hypothalamus. They're going to detect low water levels in this situation. They send a signal to the pituitary gland, also in the brain, or just outside the brain. And we're going to say that it releases lots of ADH, which is a hormone you need to know lots about. ADH stands for antidiuretic hormone, so effectively it makes you urinate less. So this ADH makes the walls of the collecting duct more permeable to water. So more water is reabsorbed, meaning that less can make it to the bladder. So resulting urine is low in volume, concentrated, yellow in colour. And if we zoom in on this bit, it makes the walls of the collecting duct more permeable to water. Let's just have a look up here. So effectively, when it becomes more permeable to water, it inserts water channels in here. So rather than the water continuing its passage down here to the bladder to form urine, the water leaves and is reabsorbed back into the body at this point, meaning that the urine produced will be lower in volume. So now we're going to make a comparison when you've had lots to drink. So we're going to say this time that osmoreceptors in the hypothalamus detect high water levels. We're going to say the opposite in the pituitary gland. So the pituitary gland releases less ADH. This makes the walls of the collecting duct less permeable to water. And remember, if you like these sorts of answers, they're all in my revision guide at the online shop. So less water is reabsorbed, and therefore resulting urine is high in volume. So we're saying the exact opposite: dilute, pale in colour. Now we're going to touch on cloning. So first of all, animals, e.g. Dolly the sheep, and I'll just give you my perfect answer here. I'm going to keep this answer super concise. So effectively you want to get the animal that you want to clone and obtain a body cell from it, and you want to remove the nucleus, which we call enucleating. So I'm going to write enucleate body cell of animal being cloned. Your next step is to enucleate an egg cell of an animal of the same species. And so effectively you've got your body cell nucleus, you've got your empty egg cell, and you're going to combine them together. And notice that in order to do this, you need to use electricity. So fuse, this is your third step, body cell nucleus with empty egg cell using electricity or an electric shock. So that was step 3. And now basically you're ready to implant this cell into the uterus, so the womb of a surrogate. And remember, a surrogate is someone that carries your baby who is unrelated to the baby. So Kim Kardashian, to give you some celebrity context, has used a surrogate several times, I believe. And then the embryo will divide by mitosis. That's worth 2 marks, so don't forget this point. And eventually you'll end up at the baby stage. But that's everything you need to score full marks. Now we're going to talk about cloning in plants, and this is called micropropagation. So your perfect answer this time is to obtain small pieces of a plant. And we call these explants, and this is worth 2 marks for saying these 2 points. And then you're going to place them, and they must be placed in sterile conditions. So sterile agar jelly. Why sterile? To prevent the growth of pathogens. And then you want to provide all the correct optimum conditions. So that means controlling the humidity and the temperature and light levels. Provide plant hormones to aid in their growth. So what are the major advantages of micropropagation? Well, it's that you can produce lots of plants quickly, and it can occur at any time of the year. So I hope I've given you lots of help with a lot of paper 2 topics. Remember that all the paper 2 stuff is in my all-in-one video, which contains both paper 1 and 2. And remember, you do need to know everything on the specification for paper 2. So anything from paper 1 could come up, as well as these extra bits you've had to learn for paper 2. So make sure you're still going over that old stuff. But yeah, here's my viewpoint of what's the most important stuff from paper 2. I'm now going to talk through how to answer that first question on the paper by going through an example. So as always, here's a huge passage which I'm not going to read out loud because that'll take forever and it'll be quicker for you guys to read it. So pause the video now to read this particular article. And now we're going to start with question 1. Explain how having few or no leaves helps the cacti to reduce water loss. Line 5. Cacti often have few or no leaves. Okay, wow, great. I love it when they do that and it doesn't actually tell you anything. Okay, so let's try and zoom in. If they have no leaves, then remember the leaf structure, which has the waxy cuticle, then it has the upper epidermis, the palisade mesophyll, the spongy mesophyll, the lower epidermis, and then remember you have guard cells with stomata. And that's the key bit here, because water loss or transpiration occurs out of the stomata. If you have fewer stomata, then you'll have automatically less water loss. So we're going to say no leaves means fewer stomata, so less transpiration takes place. Suggest how a desert plant may benefit from having extremely long roots. Line 14. Phaeotophytes are plants that have adapted to very dry environments by growing extremely long roots. Again, it hasn't told us anything. So what would be the major advantage of having extremely long roots? Well, that's so that they can reach down and obtain as much water as possible from depth. You could have also said it's for anchorage, which is to hold the plant down. Many desert trees or shrubs have very small leaves, lines 12 and 13. Explain why having small leaves would be a disadvantage for non-desert plants. Okay, 12 and 13. Some trees and shrubs are also adapted for life in deserts. They have very small leaves and thorns. Don't know why we constantly have to refer to this when it doesn't tell us anything. So it's saying, why don't you want to have small leaves if you're a regular plant? Well, why do plants have leaves? Well, it's so that they can carry out photosynthesis and produce glucose and have their own food. So if you had smaller leaves, then you wouldn't photosynthesize as much because there would be fewer chloroplasts. Suggest why having small leaves is less of a disadvantage for desert plants. Well, we know this. It's both due to decreased water loss, and really if you're in the desert, I assume that there's an awful lot of sunlight there anyway. So photosynthesis will be taking place quite efficiently. D. Seeds remain dormant in the soil until it rains. Line 22. Describe how rain enables germination to take place. Line 22. Their heat-resistant and drought-resistant seeds then remain dormant in the soil until it rains again the following year. Okay, again, it doesn't tell us anything. So why might you need water? Well, you can answer this in lots of different ways. Potentially, it could help the seed coat to split. Water also helps to activate the enzymes, and it also helps in the digestion of starch found within the seed. Give 2 other conditions that are needed for germination to take place. Remember, I used the mnemonic WOW, which stands for warmth, oxygen and water. So we know obviously they've mentioned water, so please don't write that again. They won't award you marks for that. You're going to say warm temperatures and oxygen. Suggest a reason why annual desert plants need to complete their reproductive cycle very quickly, lines 18 and 19. Most annual desert plants germinate only after heavy seasonal rain and then complete their reproductive cycle very quickly. They flower for a few weeks in the spring. And the obvious answer here is that it's because the water needed for germination is only available for a very short amount of time. Some plants, such as desert lily, reproduce using bulbs, lines 32 and 33. Bulbs enable reproduction. Small bulbs go on the side of the larger bulb and separate to produce new plants. Oh, actually, finally helpful. So if it's grown from the same plant, then it must be asexual reproduction. So that's actually quite a useful thing they've mentioned here. So the type of reproduction here is asexual reproduction because it grows off the main plant. There's no fusion of gametes. Suggest an advantage of using this type of reproduction in the desert. Well, you don't need insects or wind pollination. Name another organ used in this type of reproduction. So we probably didn't learn about bulbs when we studied asexual reproduction. We probably learnt about strawberry runners or potato tubers. So either of those would be fine. Don't know why they've given us so many lines for that answer.